Welcome back. Previous lecture we see the taxonomy and its background. In this lecture we see the types of taxonomy and the sources of data collection by the taxonomy. Let's see types of taxonomy. Types, so there are generally three types: alpha taxonomy, beta taxonomy, omega taxonomy. Alpha taxonomy, it is nothing but the initial stage of taxonomy. In that generally alphabetical like information are collected, such as the plant are identified and the names are given on the basis of the alphabet, such as the authority are given on the basis of L alphabet, genus name on given by the specific letter, species name are given by specific letter, and the alphabetical arrangement on that basis, that is known as the alpha taxonomy. Second is the beta taxonomy. Beta taxonomy, it is nothing but there is a classification in the taxonomy. In that there is a identify and name given plant. They are classified into certain biological groups, such as including the class, including family, including the into genus, including species, including the kingdom, or including the division, with the help of a beta taxonomy, assigned into specific group. And the omega taxonomy, it is nothing but the different description or the character are collected by the cytological and the genetical information. Cytological means cell information, nucleus information, or the cell organelle information are collected from that. And the genetics, that is the chromosomal or the changes in a DNA or the RNA, genetic material will be changes, that information are collected, and with the help of that description are made and the classification will be done. That is known as the omega taxonomy. Next point is the sources of data collection in the taxonomy. There are the 6 type of sources. First, morphology, cytology, anatomy, palynology, chemical, and the molecular approach. First, morphology. Morphology, it is nothing but the study of external character. With the externally we can collect the external character of plant. For example, we given a specific plant. The identify, firstly we describe that plant. How to describe? In sense of morphology. Root description will be give. What is the root description? That is which type of root present, taproot or the adventitious root are present. Another is a stem, which type of stem is present. It is a simple or branch or the unbranch. That is, a stem is a glabrous or smooth. Easily character will be identified. Next is the flower character. Flower actinomorphic, zygomorphic. Then androecium, gynoecium, or each external character of a plant will be studied, and we made a description, and that data is obtained from the morphology. Next is the cytology. Cytology, it is nothing but the cell information. With the help of that we can get a cellular data. If a plant do not having a external character, such as do not having a very much good root, we can identify them. We not having a stem, we can identify their external character, or leaf or the flower. If there is a absence of the external character, we see the next step, that is the cytological information. That means a cell information, that means chromosome number, next nucleus, their plasma membrane, cell wall. There is a structure will be studied, and with the help of structure we can get a information. That means cell information are useful for the taxonomic analysis or the plant identification. Next, the anatomy, it is nothing but the internal structure of a specific part, that is known as the anatomy, such that vascular tissue, epidermal tissue can easily identify. If the identify, we get easily information of primary growth, secondary growth. If there is a primary secondary growth is absent, that included in monocot. If there is secondary growth is present, that included in a dicot. Next is the palynology. Palynology, it is nothing but the study of pollen grain. The pollen grain detailed study are included in this branch, that is known as palynology. Shape, size, there is a exine sculpture, entire layer can easily observe of a pollen. That can be useful for the taxonomic data collection. Next is the chemical and the molecular approach. Chemical, it is nothing but special type of chemical are present in that part, that are easy for the data collection. If all these branches cannot obtain information, only 2 branches can give the information. Chemicals such as the alkaloid, phenols are the specific families are production that groups, such as the alkaloid. Alkaloid generally production generally milk E4BSA family having high yielding of a alkaloid. Next is the phenol. Phenol, it is a salty where the salty plants are grow. That condition that gives the phenolic component of the amino acid. Next is the molecular approach. Molecular approach, that is nothing but the DNA, RNA, or the any molecules are present that can helpful for the taxonomical information. That's about the types of taxonomy and the data sources collection of a taxonomy. Thank you.